So, welcome back to this course, Functional Genomics. So, in the previous lecture, we looked into the power of comparative genomics. We looked into some of the examples as to how comparing various genomes of various species help us to discover some of the changes that are having an impact as to how that species was successful. We could get insight into certain unique features. We have seen examples how you can increase the utility of a breed, whether it's a milk, whether it's a muscle mass, and so on. So now we are going to look into how really we look at the genome. Some of the examples as to how the genome has been information has been used to decipher the changes and concepts or fundamentals behind that. Let's look into this particular slide. What we are talking about is that you have had a species X to begin with. With time, this species has evolved and diverged into A, B, C, and D, right. So now, it obviously from this plot what is shown here, it is very clear that D is evolutionarily far away from A, right, whereas C is more closer to D as compared to A, and so on. So, which clearly talks about at which time point possibly these two species would have evolved into two new species. So, that is relatedness, right? That is what you are talking about. So, what we have done in this process is that we basically look at the species that are contemporary, meaning they are living today, and try to decipher as to how the common ancestor would have been. The connecting links are normally lost. We really do not have for many of the such events, we do not have an evidence as to how it could have happened. We basically look at the genome and try to infer as to how similar or how dissimilar they are, and try to plot and then sort of calculate when the species could have diverged, right. So, it all depends on what question you are asking, and that determines how you are going to compare, what kind of comparisons that you are going to do. Let us give some examples. For example, what is the core set of proteins in multicellular organism? So, if this is a question, for example, you want to ask the question, what are those proteins that are involved in, say, a very fundamental process like cell division, or for example metabolism. These are pathways that are highly conserved, where you take either plant or animals, these are very similar. So, if you look into genes that are highly conserved across species that are diverged, for example, you talk this as a human, this as a grass, and still there are genes that you can simply swap. Histone, for example, you take a gene from the grass and or take a gene from human and put it in grass, it possibly would work exactly like histone, because they have set function that is conserved for many years and frozen for whatever function they do, and therefore they do not really change. So, it depends on what question you are asking, or for example, you are asking a question, what sequence shows a signature of purifying selection that are likely to be functional. Let us assume that this particular branch represent marine organisms and this particular branch represent terrestrial organisms. So, obviously, this branch on the left, it represents certain changes that helped the terrestrial organism to survive on land, whereas here they have selected genes that help them to live in the water. So, this is what you can ask, what sequence show a signature of purifying selection, likely functional, and so on. So, you sort of look into and so on, or what sequence account for unique features of organism. For example, humans, now we have higher order brain functions, we have language abilities and others which is lacking in primates. So, what really made this change? So, you can compare with, for example, primates and ask the question, what are the unique signatures that we have which helped us to have the higher order brain function as compared to primates, right. So, that, but if you compare it with cats, then it is going to be far away, because that would not tell you the difference. It also talks about how they can hunt and many other characters that come in. So, you have to go for the close related species, yet they do not have this unique feature, and then look at what the difference is. So, this all depends on what questions you are asking. That such is the approach. Let us look in here. So, when you talk about comparative genomics, we are pretty much covering the entire spectrum of species that live on the earth. So, it goes on to this phylogenetic tree talks about the early evolution to what you have. So, we simply we have this here. So, the animals represented here, the rest are all the other species we are talking about, the primitive species that like Euglena and others, amoeba and others, fungi and so on. But if you go to more animal phylogeny, that is what is shown here. So, you have all the invertebrates here and then the vertebrates coming over here, and then of course, you have the Homo sapiens here. So, now it all depends on what questions you are asking, right, that would help you to answer. So, if you are trying to look at how, for example, the chimpanzee are diversified from the humans, and how similar they are or dissimilar they are, you are going to look into the genome sequence of these species. But you are talking about how mammals evolved from, for example, birds as compared to birds, right, or the ancestral species, then you are going to go and look at larger number of species. So, this is the way people do it. And what is interesting here is that most of the proteins that we have, humans have, are ancient. It is not that we have something unique. Majority that we have, even a fish would have, right. So, you can see here, if you look into the protein percentage in terms of coding potential, it is about only 1% of our protein apparently has no homology, meaning they do not show a similarity with other species, they are unique to human, that is 1%. In other words, 99% of the protein we share with other species. Closely 22% of the protein that we have, a small fraction again, is vertebrate only. In other words, if you combine 22 with 23, that means 77% of the genes are present even, for example, in starfish or in earthworm and so on. So, that means that we have carried the same gene, some variations in terms of function and so on, that is what it is. And vertebrates and other animals and other eukaryotes and prokaryotes, this includes even bacterium and for example, even plants and others, we share about 21% of the gene, they have very similar function. That tells us how over time our genome has retained some of the genes because they are doing the critical function which cannot be altered. But at the same time, there are other genes that either came in new set of genes or the existing genes have changed a bit and become took up the function that are either unique to your kingdom, for example, the vertebrates and so on. So, that is what happened, right. But this information has come by looking at the genome. So, otherwise you would not have told, if somebody would have told you that we share 70% of the genome with, for example, the rest of the animals, including, for example, the fish and invertebrates like octopus and others, you would not have believed, right. So, but this has come from such kind of analysis where you compare the sequence and try to classify them and tell, right. The other plot that also come from a paper is that the age distribution of the human gene families, what we look into is that when possibly the genes have evolved. So, you can see that these are all molecular time scale, these are millions of years ago, and you can see that this is possibly when this again talks about how the genes evolved. This is where the fly vertebrate split, diversified, and then this is where the animals, the vertebrates started coming to the land, and this is where the birds and mammals evolved, and mammals radiation took place, meaning they really flourished. A large number of species evolved and they sort of conquered the earth, that is what happened. So, you can see that majority of genes still are there. There are some of them are pretty old set of genes still we have. So, that is what this bar diagram again represent. This is just to tell you the molecular evolution, how whatever understanding that we have has come from the genomic revolution that we discussed so far in this course. So, what is shown here is the genomic revolution. We talk about the birth of bioinformatics, you have the gene banks, and then a large number of databases, for example, for the protein, for the and the method to analyze and compare the sequence, like for example, Clustal is one of such sequence comparison program, and then you have all these other powerful algorithms that really helped us to analyze the sequence, right, the BLAST program and so on, right. And then you have other powerful tools that have come in. For example, PFAM is a tool that identifies the protein families, and then you have this NCBA website every day. If you are a biology student, you go and look into. Then you have the UCSC browser that we have looked at often as to how really it helps us to compare the genomes of various species, identify functional elements and other ensemble tools. And this comes along with, this is in this section it is shown is the genomics. For example, the Phage T7 sequence is done, PCR is invented, chloroplast genome was identified. Then you have all these genome information coming in, ESTs, E. coli genome, yeast chromosome, hemophilia, Haemophilus genome was sequenced up to the human, right. So you have the explosion of the genome, likewise it developed, but at the same time we were able to understand the molecular evolution, meaning the possible event that took place during the evolution, right. So that's the a neutral theory, testing molecular clocks, right, substitution rates in organelles is another important thing. So we often compare the genome and tell these are the genes that are highly evolving, right, or they are evolving very rapidly, right, and these are the genes that are showing slow evolutionary rate. How do you really tell this? So if you have more substitutions, changes in the codon, especially third base, and amongst the members of the same species, right, wild species, that means that the gene is still allowing changes. What does it mean? It means that the function of the gene is not well settled, so still there is a change that is happening. Once that whatever function is set, probably it will not change beyond that, because then it becomes essential for the organism. When a gene is not that much essential for the organism, even if there are changes, that is allowed. You will find an individual having the variants. But if the gene becomes very essential for the organism to survive, if there is a change in the gene, then that individual may not survive. Therefore, you do not see the variation. So this is how people identify genes that evolve faster, or the genes whose function has been sort of frozen, right. Then you have all other such remarkable discoveries. The people talk about the fish antifreeze mechanism, even they can live in sub-zero temperature, and then duplication rates, how for example the genome has duplicated, especially in the teleost, the bony fishes, and that led to the evolution of vertebrates, right, that we will discuss now a little later. So these are some of those important discoveries or hypotheses supported by the analysis from the comparative genomics aspects, led to the newer understanding of evolution. So this figure just to show you that what are the different species that have been sequenced or are currently under sequencing. So you can see it's somewhat dated, but you can see that we are starting with bees, honeybee and other such insects genome. Of course, fruit fly, mosquito, sea urchin is again important species, because that connects the echinoderms with the fish, because that is how the invertebrates to vertebrates have evolved, and they have a fantastic genome that helps us to understand how the genome would have evolved. And there are early fishes to zebrafish, pufferfish. We will talk about this powerful animal pufferfish, because it has got the smallest genome, right. It has almost the same number of genes that humans have, but it is 1/10 of the genome of the human. So it's like a compact version of a species that is very unique, and you can understand how the genome functions there. And there are of course other species in the reptiles, some of the turtles, snakes and lizards, and zebra finch. Again, these are birds that Darwin has studied. If you can understand what is process, what do you talk about now is adaptive radiation that have come from the zebra finches. And then of course there are several examples when it comes to mammals, starting with the primitive mammal, what you call platypus, which lay egg but still a mammal, and 2 the opossum, horse, pig, and up to human, right. So, you can see that these are the variety of species whose genome is being sequenced or completed, and it gives you a wealth of information to to mine for, look for, and understand. This is only to tell you how important it is, because majority of such information with regard to sequencing the whole genome of various species have been published in journals like Science and Nature, which really talks about the importance of such genome information, including, for example, panda. That's a species is not a good model, but it is a unique species that gives us, and for example, Neanderthals, the primitive, the humans are the forerunners, right? And for example, the ancient human genome, some of the fossilized samples which represent possibly the ancestor to Homo sapiens, people have sequenced. We will talk about how, for example, even language, other evolution probably linked to this gene. We will give some examples, right? So, these are very important, but it is not restricted to the animals. It is equally important also to look at the genome of plants, because they are the primary producers. Our food mainly comes from the the plants, and we have rice, we have wheat genome, tomato. You talk about any commercial plant, we do have this sequence coming out of such species, right. So, what do you do with this? How really you do genomic or comparative genomics? So, one is that you look into, for example, the genome aspect. You look into the different species with regard to the size of the genome, right. Second, you compare the genomes and look at alignments, meaning what are the regions that are similar and what are the regions that are not similar. Third, you go for synteny, meaning you have a chromosome in human, say chromosome 1, you have a gene order in chromosome 1, say A, B, C, D, E. Now, what is that the order for these genes in, for example, in chimpanzee? Is it chromosome 1, these 5 genes are together, or they are split into many chromosomes? So, that is going to tell you how the chromosomes have evolved. So, we will look into one such example, and second is gene number, right. So, you may have the same kind of protein, but for a given protein you have at least 3 different forms of it, because the genes have been duplicated or the total number of genes have changed, right. That also tells you. And what is called as anomalous regions, there are regions that are very rapidly changing, right. The revolution rate is very high. For example, Y chromosome in case of mammals, there is a huge difference when you compare from one species to the other, whereas X chromosome is highly conserved, is not much change happening there. So, these are the information tells you what happens to a particular genome, whether it is undergoing evolution, whether it is conserved, how it could have evolved, and these are the information that come when you look at the whole genome in total. There could be also gene centric studies. So, I am not interested in the entire genome, but I am going to look into a particular group of genes. Say I am looking into genes that are involved in higher order brain function, cognitive abilities. So, obviously, if you higher order brain function or the genes are going to be enriched in organisms that have cognitive abilities. So, I can now compare the apes with other mammals and compare it with humans and see what are the genes that really makes our brain to function the way they are. Therefore, we are studying other species, right. Or go and look at gene clustering, for example, how the gene family has evolved, right. So, we will give you some examples on this. And finally, we also look into what is called as gene sequence variations. This is an important area which directly affect the humans, because for example, the SNPs are single nucleotide polymorphisms or insertion deletion polymorphism or pseudogenes, genes the sequences that have gene structure, but they are not functional. These kind of signatures are very important for the human health, because while all of us share 99.999 whatever percent of the genome sequence, the small difference that we have between individuals makes a huge difference in terms of why you are, for example, able to tolerate a flu infection, whereas I am more susceptible, right? And things like that, there are everything. So that's where now people are doing what is called as a whole genome association, GWAS, what they call genome-wide association studies, which clearly talks about the variations across a group of individuals compared to another group of individuals. What are these two groups? One group could be normal. So, for example, I am from Kanpur. I would like to understand how pollutions here induce asthma in the population, right? So now it's not that everyone is asthmatic. Some of them are still, although living in the same environment, still have not developed asthma. So it could be there are certain elements in your genome which makes you resistant for this producing this allergic reaction, right? So what is that signature? So if I have individuals from Kanpur population, one that have developed asthma as compared to the other who did not develop asthma, then I am going to compare these two genome sequences and see what are the signatures that are unique to one group, not the other. That would tell you either genes that give you risk of developing asthma or genes that give you resistance, right. So, the last section, the gene sequence variations and how the variations help in your survival is something that we will not discuss in this course, but this has been dealt with in another course called Human Molecular Genetics. I would encourage you to go and look at those videos that are available freely. If you are not doing that course, that will give you more understanding. So, right now the remaining talk we are going to talk about the whole genome and gene centric studies with regard to how they help us in understanding the evolution. So, let us ask some simple questions. Why comparative genomics? Why at all you need to do this study? This is we have discussed some examples. Still I am going to go back and ask this question because that will help you to appreciate as to the importance of this field. Conservation over long evolutionary distance suggests functional constraint. This is a statement. What does it mean? What does it mean by functional constraint, right? The functional constraint is that your genome changes, right? Your sequence change, and the change is not directional, meaning the process does not identify a region and changes here. It is random. It happens because of the mutation. When the DNA replicates, when the copying machinery is not faithful in copying, or when the repair machinery is unable to correct all the errors, some regions you introduced some new change, and this new change, whether it would come to, when I say introduce new change, it happens during the meiosis. In the meiotic cell, that is one come to the next generation, and if so happen it became a gamete that fertilized an egg, whether the new change would come in that individual, whether the individual would survive, depends on whether the change has affected the individual or not, right. So, suppose that the change did not affect the survival of the embryo and the birth of that individual, then you have seen that mutation in that individual in the population because that guy comes in. So, now you may have a problem. For example, you may have a sixth finger, that is because of a mutation. Now, the sixth finger is not affecting his survival, he is as good as any one of us. So, you would see them in the population, right, that mutation has come in. Some mutation would not affect anything, he is absolutely normal just like you, right, and then it is there in the population, it does not really affect anything. So, it is called a neutral variation. Some could be someone is born blind because of the mutation affected the way eye functions. So, he may not transmit this gene to the next generation because he may not marry. So, it stops there. Some affects even the embryonic development. So, that is aborted, the embryo is aborted. So, the process of introducing new variants in the genome is a random process, but whether that new variant comes to the population depends on whether the variant has affected the function of the gene or not affected. So, if you look into the normal population, what you are looking at is variation that are allowed because they did not affect any of the gene function. So, obviously, regions that are not very critical for gene functions are the regions that have more variation because it is allowed, because you are normal otherwise. So, that and the opposite of it is that there are regions in the genome in which variations are not allowed, meaning if variations happen it affects the gene function, therefore, the person may not survive, may not transmit the gene to the next generation. Therefore, there may be some regions where there is a constraint, there is a pressure not to allow changes, meaning by natural selection, right. So, that is what you call as the functional constraints. So, that tells you which segment of the genome is having a critical function. So, if you compare a large number of genomes of different species, then you will find segments that are showing higher homology, similarity, but not they are genes, they are not genes. Then that gives you information that this segment has got an important function for the cell, for the organism, therefore, it is not allowed to undergo changes, right. So, that is important information. Lack of conservation over short distance may indicate of adaptive evolution. What do you mean by adaptive evolution, right? So, you must have studied about mammals and other Darwinian finches. These are classic example adaptive radiation. When there is a pressure, like if for example, the same population of a species, members live in a locality or habitat, if there is a sudden pressure on them, change, right, then what they do? They migrate to different region to survive. So, they wherever they go, they may find a new challenge. So, whether the individual of that particular species, members of that species that went there, whether they were able to survive in that new place depends on whether you have had some variant that made you successful over there, right. For example, it could be that you were driven away from your habitat, you went to a place where it is a region where it is close to the waters, right, seashore kind. Then you have to have abilities to sustain that kind of humid condition. Or you may go to a desert region where there is no water at all. So, you have to have adaptation or some abilities to survive with little water, or you have gone to higher altitudes where it is much colder. So, you have to survive that cold temperature. So, if you had some changes which otherwise were not helping you in the original place, since you are driven out now, if you go there, now that particular change that you have which did not really help you earlier may give you an advantage, right. So, you die and then you populate, you make a colony. Now, if you isolate these species that originally diverged from their common ancestor very recently, and then you do a sequence analysis, you will find short regions of the genome that show lot of variations. That talks about how the adaptive evolution took place. That is another example. Helps us to identify both coding and non-coding genes of unregulated elements. Again, it is an important aspect. The coding segment is we told you less than 1%. The remaining is regulatory regions. How will you identify what are the regulatory regions? Sequence wise is going to be extremely difficult. So, if you compare the genome, probably it will give you a clue as to what are the segments that are highly conserved, which gives you an information as to these are important elements in functioning the gene or its expression or any of the other aspects. We will give some examples as to how people have understood that. Characterizing the difference between organisms reveals mechanism of change, right. So, it tells you like how certain organisms are successful in certain environment, right. So, it really tells you like how certain species are successful and what are the changes that help them to be successful, and you can understand the vertebrate evolution. So, all of us from fish to the mammals, we all evolved from a few set of species. So, that is why we share the genome, and we want to really reflect and see how we evolved. So, that really helps. We will show some example. Knowledge between species for annotation and inference of function, like we told you, gene ontology is another example, where we are studying yeast, understanding a function of a gene. That gene we also have. So, that information helps us to understand what is the function of that gene in the human, and tells us what is common and what is unique between different species at the genome level. So, for example, why humans are very similar to apes, we are very different from apes, right. It tells you most like we are successful because both of us can function, walk with leg, bipedal, only 2 legs we can walk. So, that made us to use our forearm, which otherwise we are using it for walking. Now, we are able to use it in a different way. So, we can make tools now because no longer the hands are used for walking. So, it was a gift given by the primates, and this thumb we are able to put it across the 4 fingers. Again, it helped us to hold the pen or chisel or whatever it is. We are able to make tools because of this. Again, this is a gift given by the primates to us, right? And then we have developed our own qualities. For example, our brain is far more functional, more sophisticated, cognitive abilities. That is beyond what the apes can do. So we have acquired something new that makes it unique. So we can look into these gene signatures, and the function of human genes and other origins may be revealed by studying the counterparts in simpler model systems. For example, if we cannot understand the gene function in human, we can take this sequence, engineer the animals, put it over there, and understand what could possibly be the function. So we are going to look into some of these examples, how the model systems we were able to use to understand the human gene function. So let us look into one good example, that is the platypus. Okay, this is a sequence, this is a Nature issue that carried the genome sequence of platypus. What is platypus? Platypus is a mammal, monotremes, what they call primitive mammals. You can see that these are called as, you have amniotes. The amniotes are those in which the embryo grows in the amniotic fluid, whether it is birds or reptiles or mammals, we all have amniotic fluid. That is why it is called as amniotic fluid. And then you have these two lineages, that is birds and reptiles. Again they have egg, but they are amnion and amniotic fluid. And then you have this mammalian lineage, in which you have these prototherians, primitive mammals, which includes, for example, the monotremes example is the platypus that we are seeing here. They are egg laying mammals, okay? They lay egg, but they still they are mammals. And then you have what is called as the marsupials, which are kangaroos, which are again they do not have egg, but the newborns are not fully developed like humans, right? So therefore they remain in the pouch, they develop and then only they come out. And then you have this eutherians or true mammals, in which the live young one is completely developed, so that's the young one. So you have these categories. So there were theories which suggested that this platypus, what you are talking about, could possibly a connecting link which connects us with the reptiles, because they have egg and they have skin like, but they have the scale and things like that, right? So that's what was the hypothesis. Therefore, and also they have venom. This is another important thing. So they are just like the snakes and some of the lizards. They can spike venom, which can be lethal, right? So that they have. So therefore, there was a one theory that they are more closer to the lizard. Therefore, they are connecting link, between reptiles and the mammals. When they sequenced the genome, then altogether you found this kind of a lineage, which was unexpected. That's what it is. So where they are, this platypus lives in the eastern part of Australia. So this is the region that you find platypus, not in other parts, right? And what is unique about the platypus is that there are two ways by which they fertilize the egg grows. One is that once the egg is fertilized, the division is unequal, like what you see here. So you have one smaller half, one bigger half, and go on developing. This kind of cell division you find normally when there is a yolk, for example, chick and many other, which are characteristics of the species that are present on the right side, the reptiles and birds and so on. Whereas mammals, follow this equal, cleavage. So one cell divides into 2 and 4, each one of same size. That's why when at times when the embryo splits into 2, then you will have identical twins. So it is possible because all the cells are equal in size, unlike in case of majority of the reptiles and birds. What is unique is that platypus follow this model, that is, they have a cell division in which one cell is larger than the other when this division happens. Therefore, by looking at the embryonic development, people always classified this is closer to the reptiles as compared to the mammals, right? So when they have done the genome sequencing, it was understood that they are like what is shown here, that the platypus is more closer to the mammals than to the reptiles, right? We can see here they are separated probably about 315 millions of years ago, and you have like what is called the mammal-like reptiles, right? Though they are mammal, and they are the common ancestors for all the 3 major groups of mammals: monotremes, marsupials, and eutherians, right? So it's important to know that both monotremes and marsupials, these are unique to the Australian continent, right, whereas eutherians present everywhere. So, but we do not know. So, but obviously they represent a different lineage about 166 million years ago that diverged the prototherians and therian mammals. Again, you have what is called the holoblastic cleavage. This is what I am talking about. Possibly this species sort of evolved a system wherein the egg is divided into two during development. This also could have happened because the egg no longer require a yolk, because the embryo is bathed in amniotic fluid, and the fluid is always nourished by the mother. Because of that, it need not have such unequal cleavage. So, the placentation and possibly the growth of the individual within the mother has helped really to go into a different mode of development. That is what you call as a placentation viviparity, right. So, these are some other points that really explains. And what is interesting is, then if that is the case, and the platypus has got venom, this is the tip of an adult platypus spur somewhere close to the hind limb, the lower part of the leg. So, you have this spur which can eject venom, which can be very, very toxic. And what they have done is an interesting study. They have looked at the evolution of the gene that codes for the venom. What is interesting is that there are different lineage, and all these venoms are proteins, nothing but these are beta defensin, what they call lineage, a group of proteins. It is not that it is closer to the reptiles, because they have you have the snakes and they have this venom. Again, these proteins are more closer to the mammals, and we also have this same protein defensin, which the platypus use as a toxin. But here it is much milder in our body, which is secreted, as I told you before, in the intestinal lining and in the skin, which helps us to prevent bacterial infection and so on. So, it is only the the way the protein is evolved in a given species made it more toxic, whereas here it is lesser toxic. So, that is what it is, but it is what is called as a parallel evolution. It is not something that they acquired from the lineage that led to the reptiles. Now, let us look into how the evolution takes place, okay, the speciation. So, how is that happening? So, let us say that this is species A and species B, right, and these are the contemporary species that you see today, and probably they evolved from a common ancestor that is shown here, right. So, first of all, the question is, why should they become two different species, right? There are several possible events that leads to new formation of new species, but I am just showing you one. One could be geographical isolation. So, these species now they migrated, say, on either side of this the hill and lake. Now, this particular species now they remain here for a long time. So, they are exposed to very different kind of environment. Likewise, this species remained here for a long time, they are exposed to different kind of environment. As a result, there are different changes in the genome that were selected to make them successful here as compared to the other species. So, over time, if they are not breeding together, therefore, there is no gene flow, then each would acquire a unique set of genomic signatures. Now, they cannot breed, have F1, therefore, they become an individual species. That is what we talk about. So, if you now look into these species and their genome, we can understand now. So, this is what it is. They are together, this is the ancestor, they both belong to the same lineage, but evolved over time. Now, we are looking at this is, say, for example, species A and species B, and we are looking at a given gene, and these are the exons, two exons that are there, we are comparing. Now, what is shown here is the percent similarity. So, you can see here when it comes to the exonic region, you have a higher similarity, but it goes in intron, again you have exon high similarity, it goes down. What does it mean? It means that in the intronic region, with whatever number of generation in between before they became individual species, there are a lot of changes that happened, and these changes did not affect the gene, or probably it helped in the species to survive in that particular habitat, right. Therefore, it diversified, but the coding region still have higher similarity because there is a pressure on what kind of amino acid that gene should code for. So, the changes are not allowed or they are not selected, therefore, you do not see in this population. Let us look into something else. Say suppose there is a region here, right, that region is very critical for the gene function. It could be an enhancer. Remember we said that there are elements within the gene on which some protein comes and bind and then activate the transcription, help in the activation of the transcription. Now, if there is an element and that element is certain DNA sequence, right, and if the sequence is altered, the protein cannot come and bind. Therefore, there is a pressure that that sequence is not altered. So, if you all of a sudden you find 2 exons which are very highly conserved, plus there is a region that is highly conserved, that immediately tells you that this is a regulatory sequence. It has some specific function. So, this is how we will try to look into the genome, and that is what it is. So, basically you look into the sequence. If you look, for example, human and mouse, the sequence looks like this and you may not really make any difference, but if you analyze very carefully, you will find certain signatures that are identical in these 2 sequences, which tells these are the reasons possibly there is some functional significance, therefore, they did not change much, right. So, that would help you to understand further. So, I am giving you some example. This is a gene called DACH. Let us not worry about what is the gene, but what is interesting here is that we are comparing from fish to mouse a small segment of the gene is the core enhancer element. Okay, it is present in one of the intronic region, and this is not coding for any protein. Therefore, there is no constraint on protein sequence, but it is purely the base sequences, which is 98% identical for 350 base pair in humans, mice and rats, approximately 1 billionth of parallel evolutionary time that separates human and other fish. You find that this 220 base pair fragment remain frozen, they did not change at all. That clearly tells this sequence is so critical for the gene function. If there is any change, that would not allow the organism to survive, so you never see it in the population. So, always you find this sequence to be present, which clearly tells that it is a critical region, not coding for any protein, but still it is very critical. So, this you would not have understood if you have not compared these many genome sequences. That again gives you a glimpse as to how comparative genomics can help you. So, you can go and look at I am sure you would have seen couple of lectures by my TAs where they have given you some introduction on how to use various bioinformatic tools. You can go and analyze the sequence, you should go and try it out. This one of them is the University of California the Genome browser. We have introduced many a times in this lecture. So, you can go and look at any region. So, again I am giving you one particular gene sequence, right. So, you can see this is a UTR, right, untranslated region, and we are finding it, there are regions that are highly conserved, right, and these are the regions that are, regulatory elements, some proteins comes and bind, so that you can see that you can compare with a large number of species, you find that that region is present, right, this sequence you can see here. It is very, very, very critical for function. So, this is information that you can get by comparing the sequence, right. So, we will stop here, and then the next lecture we will see a few more examples as to how you test this hypothesis. That is, if you have identified an element which you believe is a regulatory element because it is conserved, how will you test whether indeed it is doing that function? See you again.